Yeah, thanks so much for inviting me to this birthday conference. This is super fun. Yeah, so happy birthday, Mike. With some kind of 6 months delay, I guess. I used to be a string theorist, and you know, Mike and I used to work together here at this is the Simon Center in Stony Brook that I guess many of you know. And so this was back in 2008, where the Simon Center was founded, and Mike was the first member at the center, and I was the first postdoc with Chris Beasley at the time. And so I wanted to mention a few episodes of our interaction with Mike. And so at the time when we moved to Stony Brook, the Simon Center was not there. And in fact, the Simon Center was just the 5th floor at the Math Tower in Stony Brook, where we used to have our offices. And so for a few years before they built the building, we were hanging out there. And so my first interaction with Mike, I don't know if Mike remembers that, but my first interaction with Mike was about a project. One day he came to my office and he was like, "Oh, so I'm working on this kind of cool compactification idea. I'd like to calculate the effective potential for the warp factor. And I think we can use the Hamiltonian constraint from general relativity to come up with some kind of Schrödinger equation for the warp factor and effective potential. So are you interested in this?" And I was like, this sounds super cool. Yeah, but I never really studied Hamiltonian constraint before. So what I did is so Mike suggested, well, why don't you look at Wald? There's like an appendix in Wald where they talk about it. So I was like, okay. So I started to read the appendix in Wald. I thought it was kind of super fun. And then what happened was that a week after our first interaction, Mike came back to my office, was like, so you remember that thing we talked about like last week? So actually, I finished calculation. I wrote a paper, so I'm posting it on the archive. It's like a 100-page paper. I was like, oh, okay, that's awesome. The paper. That was kind of cool. And so I was like, maybe I should do neuroscience. And so then actually, yeah, but that's what I did. But before doing that, then I ended up with this super fun collaboration with Mike and Shlomo, where we wrote this nice paper on the holographic dual of free field theory, which was really, really cool and technically very challenging because we proposed, the higher spin Vasiliev theory as a dual, and so it was kind of a super fun collaboration. So that was so this is the paper I wrote with Mike, and I wanted to mention that. This was like 2011. By the end of 2011, I made up my mind to move to neuroscience, and Mike was incredibly supportive with this. And I still really cherish our conversation. And it was super interesting to figure out that actually that Mike was very, very interested in artificial intelligence at the time. Once I mentioned that I wanted to do neuroscience, he was like, "Oh, that's amazing!" Which was what changed my, kind of the way I did research. He was like, "Well, what if you really want to do neuroscience? Why don't you take like a few months off, our string theory, and spend time to create a course on computational neuroscience that you can teach at the Simon Center for our graduate students and postdocs?" And this was like amazing. So that's what I did. I spent like a few months like studying books and papers like classics in computational neuroscience, starting from the Hopfield network and that kind of series of works. And I prepared this class that I taught in the fall term in 2011, I think. And it was very well attended by all these string theorists that somehow were interested in learning about neural networks. And that's what like totally totally convinced me that that's what I wanted to do. And then in 2012, when my appointment at the Simon Center ended, I moved to the neurobiology department, and I started to work in neuroscience. And so, yeah, so this was like some kind of amazing interaction that I had with Mike, and Mike was so supportive, and it was great to hear that he was very, very interested in artificial intelligence. And then so much so that many, many years later, then in 2020, we got back together after, you know, I spent like 8 years doing neuroscience, and I started my lab at University of Oregon. And so we got back together, and we organized the first neuroscience or AI conference at the Simon Center. And so this was like one of these programs that the Simon Center is running, so Neural Networks and the Data Science Revolution, and it featured one workshop on string theory and data science, and one workshop, which is the one that we organized with, that was on the physics of neural circuits and network dynamics, which was super, super fun. And some of you in the audience were attending as well. And so, yeah, so this was kind of coming back and interacting with Mike so many years after we last interacted in string theory. So, yeah, so this is to say that, yeah, Mike has been an amazing mentor for me, and he's been so supportive when I decided to switch into studying neural networks and neuroscience. That, yeah, so it wouldn't have been possible without his support. It's pretty clear to me. So, yeah, and so today I wanted to talk a little bit about the work that's been done in my lab. So this is my lab at University of Oregon, where we have an Institute of Neuroscience, and I'm blessed to have a super fun group. Many of the trainees in my lab are actually recovering theoretical physicists. In fact, all of them are physicists, and Merav is a former string theorist, and Alireza is also a former string theorist. And so, yeah, so I know, I keep interacting with theoretical physicists, because that's kind of the mindset that I have. And so today I'm going to talk about a theory of how naturalistic behavior could be generated in the brain. And in my lab, we have many projects that cover different aspects of this. And today I'm going to talk about a couple of them, depending on the time, I guess. So I start with like a cute video, because all we do in the lab usually, you know, this is neuroscience. It's not string theory. So we look a lot of YouTube videos to draw inspiration on how the brain works. And so here you can see a YouTube video of squirrels that learn how to solve a super complicated maze to get some food. And this is to show how complex the features of natural behaviors can be. And in particular, what you can see is that, so I want to raise your attention into the kind of structure the natural behavior exhibits. And so there's like very, very fast snippets of movements that we can call the syllables of behaviors. So these are actions that are performed at 100 millisecond scale, which include like moving to run, moving your snout, looking left, looking right. So these are like super fast movements. But there's like a grammar such that animals can combine these very fast actions into behavioral sequences. And so behavioral sequences that have a meaning for the animal, because they're geared towards reaching some goals, which is, for example, locomotion, running, like carrying a nut where you're moving through these pipes, fighting, these kind of things. So this happens at a longer time scale, about, you know, a few seconds. That's a behavioral sequence typical length. And then there's like some kind of overarching, much longer time scale where you combine these behavioral sequences in a very complex way to solve this maze. So you have to, every piece of the maze involves some behavioral sequence that you have to perform to solve the current task. But your overarching goal is to, yeah, no animals were harmed in making this video, I guess, because the squirrels can fly. Yeah, so you combine these sequences in these overarching plans or long-term goals, which is they must get these walnuts. They can last up to minutes or even hours, or, you know, for humans, many, many years, a whole lifetime. So this kind of, so the idea is that, so there's this kind of very complicated hierarchical structure with very complex temporal organization, underlying behavior. And we believe in neuroscience that this is generated by some kind of recurrent neural network up in your brain that must be able to drive all of the aspects of behavior. And so the question is very complicated because not only does the brain generate behavior, but the brain also reflects the backdrop of behavior with proprioception. So it's a complex interacting system between neural activity and behavior, and that's kind of what we try to untangle in neuroscience. Okay, so I guess enough with the fun videos. So after watching this video, I was like, oh my God, let's try to figure this out. So, so in this talk, I'm gonna try to answer a few of these questions. I'm trying to pose some questions and then answer. Actually, so I also wanted to mention, so if you want to interrupt and ask questions, please do. I hope I can. Are there questions?
Yes. There's no questions. We will interrupt if necessary.
Oh, thank you. Thank you. Okay, so here's the question. So how does the brain generate this temporal variability in actions? And we'll quantify the temporal variability and see exactly what it looks like. How does the brain concatenate these actions to create these behavioral sequences that are geared towards a goal? And once you have this kind of structure in place of generating actions and concatenating them into behavioral sequences, how can you flexibly control these sequences, in particular the speed of these sequences to kind of adapt to different contexts or internal states? And then eventually, how do you actually put together these different timescales into like a hierarchical structure that spans timescale from 100 milliseconds to several minutes, like over many orders of magnitude? So this is kind of the kind of questions that we think about in my lab. And so, so I'll give you kind of the big picture and kind of the answer at the top. And so I just wrote a review on this if you're more interested. So this is the kind of the summary of the neural variability of the variability in behavior here on the left. You see it. So there's this hierarchy of timescale from milliseconds to minutes. That is one axis of behavioral variability, if you want. At every level of these timescales of this behavioral of this hierarchical structure in time, so one can modulate the typical timescale of one behavioral units, for example, of an action sequence or a movement, depending on the context. So if the animal is distracted or is attentive in the zone, they can modulate their reaction time to stimulate or the time it takes for them to perform a behavioral sequence. And this happens at every level of the hierarchy. So this is a modulation. It's kind of a second axis of temporal variability. And the third axis of temporal variability, which I call a stochastic axis, is what's left after we account for all the known sources of behavioral variability, which include this hierarchical structure, the context, and other individual behavioral differences, phenotype differences in phenotypes between animals. What's left in a single animal in the same context, it's still a huge variability in the repetition of the same action, for example, which is of stochastic natures. And I'll quantify this this soon. And so I think these are like three independent or roughly independent axes of behavioral variability. Yes, question? No. Okay. So these are roughly three independent axes that we can try to study using our tools in neuroscience. And a potential answer for how these different axes can arise in behavior comes from a theory of attractors, which I think maybe Remy talked about earlier in his talk. I'm not sure from the abstract but it might be. So there's been an emerging consensus in the last few decades, starting from the Hopfield network, but more recently with more experimental work that provided kind of proof of principles that attractors exist in cortex. I think that attractor dynamics can actually explain or attempt to explain all of these three different axes of behavior. And in particular, one can study, so I will study, the first case I'm gonna present is how attractors can explain the stochastic variability of single actions and how these are concatenated into action sequences through transitions driven by noise in some brain area. And then once we have the structure with attractors, which are just potential wells in a landscape, if you want, then we can modulate the transition times between attractors by changing the depth of these wells. And this could explain the contextual modulations using a technical term in neuroscience I call gain modulation. It's a modulation of these potential well barriers. And then by the end of the talk, if I have time, I'll show you that, well, I probably won't have time, but yes, I'll just mention here. So one can also explain the full emergence of these temporal hierarchies within this attractor picture by assuming that there's neural assemblies of different sizes in the brain, and the larger the size of this neural assembly, which is an assembly of many neurons, the slower its dynamics. And so by positing that there's this kind of different heterogeneity in size of cell assemblies in the brain, one can show that this can lead to a hierarchy of timescales to emerge from the same cortical circuit. So the idea of having kind of attractors, attractor dynamics can in principle explain all the features in behavior along these three axes that I mentioned. So this is kind of the framework of the work we do in the lab. And okay, so for now, I'll just focus on this first part, which is how are actions represented in the brain, and how does this variability in the repetitions of the same action arise from attractor dynamics. So in order to study that, one has to actually model behavior, which is extremely complicated, requires a lot of computer vision, deep learning, and this was pioneered by Bob Dada's lab at Harvard. So where he showed that from videos of the animal, like the videos of the rodents, the squirrels that I just showed you, using deep learning, you can classify the animal behavior. You can extract actions from these videos, and he showed that spontaneous behavior, spontaneous animal behavior can be understood as a sequence of actions where you have many syllables, like hundreds of syllables, and these syllables like reoccur at random times. And for example, this blue one, like the dive syllable, when animal dives, reoccur in different positions in a behavioral sequence, and every time it reoccurs, it lasts for a different amount of time. So there's a huge source of variability in natural behavior, which is we have many actions the animal can take at any given time. We can combine these actions in principle in a combinatorially large way. And at the same time, every action can last for a very different interval of time every time it's repeated in a sequence. And so in particular, the distribution of dwell times or the duration of each action is very skewed. So it looks almost exponential, which suggests there might be some stochastic process underline its generation. And so this sounds like an impossible problem. Oh, is there a question? No, this sounds like an impossible problem to solve. So what we thought was, well, maybe we can fix the lexical variability and still retain the temporal variability, which is what determines this kind of stochastic process. And so that's what we did with Zachmanen, in which we attempted to have a naturalistic task where the animal was self-initiating some behavior, so that every action would be self-initiated, so retaining this large variability in execution time. But the animal was performing the same sequence of action in every trial. And in fact, this allowed us to make progress and to achieve like a dictionary between each action the attractor was making, each action the animal was performing, and the underlying attractor in neural activity. And so this is work done by Stefano and Ulysses, or two postdocs, and collaboration with Zachmanen's lab, an experimental lab in Champalimaud. So this is called the marshmallow task that some of you are familiar with. This is from the psychology literature. So in the marshmallow task, there was this kid in a room, and the kid was sitting in front of the marshmallow. He could either sit and not eat the marshmallow and kind of restrain for like a long time, and then afterwards he would get a second marshmallow, or he could just like have it, and then he wouldn't get a second one. So this is the rat version of that, in which a rat initiates a trial by poking into a wait port, and then the rat can either wait patiently for a very long time until he hears a tone that's drawn randomly from a distribution, and then he can poke out and poke into a water port and get a large reward. Or this is the patient condition. Or if it's impatient, it can just break out at every moment and still collect a reward, but a tiny reward. So if it's patient, it gets a large reward. If it's not patient, then they get a small reward. The crucial thing is that the sequence of actions performed in all conditions is the same, although the timing varies. And so this is a video of the experiment, kind of blurry, but you can see the rat that's poking into the wait port to the left and then collecting the reward on the right. So this is poking into the wait port, collecting the reward, and he goes back and forth. And then sometimes it just hangs out in the back. It does other things. But this is kind of the simple sequence of actions the animal is making. Okay, and so what we're gonna answer with this experiment is, how does the brain represent the intention to act, how it generates action sequences, and how does this variability in action timing emerge from neural dynamics. So the way we do this, for those of you who are not familiar with how neuroscience works, we collect the neural activity using electrophysiological recordings, which means we're lowering an electrode in the cortex, in a particular area in cortex that's called the secondary motor cortex, like a frontal area. And you collect the electrical activity around this electrode. This is kind of some electrical activity. And then you do a high pass filter of this, and you find some events, some discrete events that occur very, very fast. These are called spikes. They typically last for a millisecond or so, a few milliseconds. And you can identify the shape of a spike around this electrode. So these spikes have different shape, they represent different neurons. So doing this, it's called spike sorting. One can extract from a bunch of electrodes, can identify the activity of a few neurons simultaneously recorded. These are like 4 neurons. And what we're gonna focus on is the spikes emitted by each of these neurons. So these are spikes are like point processes that you can see that I represented here with vertical bars. So the red spikes are the spikes from this neuron, the green spikes are the spikes for this neuron, and so on and so forth. They're all simultaneously recorded. So this is how we access neural activity in the brain. And what they did in the experiment, Zach and Mazza, is they recorded a bunch of neurons from the secondary motor cortex here. And these are like 12 neurons simultaneously recorded during a trial. And so every line here represents one neuron. Every black dot is a spike or an action potential emitted by one neuron. And we have 12 of them. And I overlaid here. So here I'm showing you 3 trials where the animal was poking in and poking out, and then poking into the water reward port with these arrows on top of each of the 3 panels. And so the actions occur at random time because the animal was just like, performing this action sequence whenever they wanted. And what we did is we segmented neural activity using a hidden Markov model, which is a way to extract some population states from these 12 neurons. And so the idea is that the way dynamics is generated in cortex, at least in secondary motor cortex, is that there's periods where neurons fire at a constant rate. So where spikes are drawn from a Poisson distribution with a constant firing rate for this kind of extended periods. Every one of these periods is color coded with a different color. And so this is like the light green, the cyan, the green, the purple. So these are all periods of piecewise constant firing rate. And then between one period and the next, the cortical circuit is switching super fast to another period of piecewise constant firing rate. So there's a question? No. Okay. Sorry, I hear voices. Sorry. So long story short, so there's so in this talk I'm kind of giving you kind of the fun results, and there's a lot of technical details, excruciating technical details, I would say, but they're all in the papers. So yeah, I'd be happy to get into details if you want. So it turns out that in every single trial in this task where the animal was performing the sequence of actions, neural activity in secondary motor cortex went through this sequence of patterns extracted with the hidden Markov model. So these are 3 trials where the animal was doing this task, and you can see that the sequence of neural patterns or neural states is the same across all trials. And so these are color coded in the sequence. And what I can do is I can take every one of these trials with their sequence of colors, which represent this, each color represents a different neural pattern. And I can just plot them here. Every line is a color coded sequence, and we have 180 trials. And you see that basically every trial has the same sequence of neural patterns. And if I stretch them and I align them to the actions that the animal is performing, poking in, poking out, poking into a water poke, you see that this is the same sequence again in all trials. And the crucial thing is that the onset of these attractors predict when the animal is going to perform the next action. So what we were able to do with the classification analysis is we were able to establish a dictionary between which attractor is coming online in secondary motor cortex and which action the animal was going to perform next. And so we can train a cross-validated dictionary during these correct sequences. And then what we can do is to then go and look at the rest of the experiment where the animal was doing other stuff, like poking in and out kind of a few times, then hanging out in a corner. So we were able to predict what the animal was doing in the erratic periods of the task of this experimental session by using this dictionary that we compiled during the correct sequences. So the idea is that now we're gonna interpret these neural patterns we found in secondary motor cortex as attractors. And we're gonna build a model for this that I'll show you in a second. And each attractor represents the intention of performing the next action. And so So in particular, so then we can interpret neural activity in secondary motor cortex as like sequences of attractors. And the important thing is that although the sequence is conserved because the animal is performing the same behavioral sequences, the duration of each attractor varies from trial to trial, just as the duration of the action, these action intervals vary from trial to trial. And the distribution of these attractors is exponential. It is right skewed, looks like exponential with the average time that's like about 500 milliseconds, which is much longer than the single neuron time constants, which suggests this might be an emergent property of a neural network and not a single neuron property. And so what we did next as theorists is that, okay, so now we have a network, secondary motor cortex that generates reliable sequences of attractors with large variability in waiting time. So what's the simplest neural network that can generate reliable sequences where the timing of these patterns varies with the very right skewed distribution of dwell times. And so what we do is we use recurrent neural networks to model this neural activity in secondary motor cortex. What's a recurrent neural network? Well, I think Remy maybe already mentioned that earlier. So it's a coupled system of first order differential equations where the variables in these differential equations, U represents the activity of neuron I. And so the rate of change of the activity of neuron I depends on the activity of all the other neurons through some transfer function, a sigmoid. And the crucial ingredient here is the couplings between neuron I and all the other neurons in the network. And so the choice of coupling determines the dynamics of this network. So for example, if we choose couplings that are a symmetric matrix, which is a bilinear, a low rank symmetric matrix that's a bilinear in some vector psi mu, then this is called a Hopfield network, and it stores stable attractors such that the psi mu's are the values of the stable attractors in the network. So we can code attractors in this network in this fashion. This is like the classic model. And what it means is that for 3 representative units here, if I start from random initial conditions, then they're gonna just converge very fast to the closest attractor with the largest overlap with the initial conditions. So this is kind of a simple attractor network. And so what we did though is that, you know, this is not good for us because, okay, we generate attractors, but we want sequences of attractors. And so in the literature, what people did is that they added 2 terms in the coupling matrix. The first term is kind of a Hopfield term, this kind of that establishes attractors as the states of stable states of this network. But then we add a second term that's called an asymmetric term that couples attractors mu to attractors mu plus 1, and it generates a sequence of attractors. So if we do that, what I'm drawing here is the overlap between neural activity and one of the particular attractors. Every attractor is a different color. We generate a sequence of activations just like this, which is a good starting point. Although what happens is that in every single sequence in this network that we generate is identical. So it doesn't retain the temporal variability. And so long story that we added a scalar noise term in front of this asymmetric term in the coupling matrix. And this actually generated correlated fluctuations in the direction of the attractors. And what this did is it generated a reliable sequence of attractors with stochastic transition times between the attractors. And so this is the picture of what the model does. Every attractor is a potential well. And so we have to generate a sequence, but we have to make sure that the transition between the attractors is generated by noise because it must have this kind of exponential distribution of transition times, typical of a diffusion process, escape from a potential well, if you want. And so what happens is that if you simulate activity from this network, you see that in fact, it generates these reliable sequences. This is what I'm showing here, three examples of trials from this network with different initial conditions. And so the sequence is always the same, but the transition times now are exponentially distributed. And so this network is actually capturing the full dynamics of secondary motor cortex. And you can show that this network is equivalent to a biologically plausible network that explains cortical thalamic connections. So this is like an effective theory for a cortical thalamic loop, which might actually generate the full brain dynamics in this. Generate these pattern sequences in this task. So, okay, again, long story short, this is a very complicated technical story, but I gave you the highlights. So we represented actions as attractors. We concatenate actions into sequences using this low dimensional correlated noise. And so the question is, why do we have timing variability at all in the brain? Why don't we just perform these action sequences, with the best timing, right? So that's required. And so, okay, so this is an attempt at a normative explanation for that. So for example, these are like bears that eat salmon that you can see, unfortunately, here in Oregon. So this is like grizzly bears. They're not very friendly, but they're very good at eating salmon. And they have perfect timing in this super complex behavioral sequence that they have to keep track of the salmon and then snap it. But it's not like they're born like this. So when they're young, they just they don't know the correct. They know what they're supposed to do, they know the sequence that they're supposed to perform. But it takes quite a while for them, maybe like months, to actually learn the exact timing of the behavioral sequence they have to perform. And so maybe this kind of variability in generating behavioral sequence timing is actually what you need to learn the correct timing. So maybe this is the reason why we have this behavioral variability, among other reasons, like, if you're a fly, you want to escape your predator and so on and so forth. But in this case, this could be a way to exploit variability to learn the optimal timing. Okay, so then once we have these behavioral sequences or this kind of action timing, I wanted to go to the next step in the diagram, which is, okay, animals can flexibly modulate. They have access to the timing of these actions to adjust their timing to kind of changing environmental conditions or to their internal state. For example, the reaction time in some task is like accelerated. So the performance in some tasks that requires some reaction times is faster if you expect a stimulus to occur, as opposed to when the stimulus comes by surprise. So there's some contextual modulation of reaction times, if you want, and of performance depending on the internal state that the animal is in. And so now we're going to use this idea of contextual modulation to look for an experimental test of this theory of attractor dynamics. And so what we do is we move from this kind of abstract we showed you before, which is the same flavor neural networks that people use in deep learning, I guess. We're going to make a biologically plausible implementation of that in terms of machinery that's in the brain, which is excitatory neurons and inhibitory neurons that emit spikes. So this is not, so the variables in this network are going to be actually spiky neurons as opposed to rate units of continuous activations, like in the previous part of the talk. And so you can generate this metastable attractor dynamics using these biologically plausible ingredients that you can find in the brain, like excitatory inhibitory neurons that emit spikes. And this is like an example of spiking activity from 5 seconds of a network of 2,000 neurons, where excitatory neurons are in black and inhibitory neurons are in red. And I arrange neurons according to clusters. Now, the important thing in this network is that neurons are arranged in clusters with preferential connectivity, such that neurons in the same clusters are very strongly recurrently connected, and neurons between different clusters have depressed connections. So there's kind of this spatial structure in this network with clusters of neurons, which is what actually, so evidence in the brain suggests that this is the case. And this suggests this metastable dynamics where each clusters switch up and down from active to inactive. And you can see the black parts are the active ones, the white parts are the inactive ones. So there's this kind of heterogeneous dynamics. And we can, like have a cartoon of one cluster that switches on and off, and so on with some intrinsic timescale, tau. And so what we can do is we can modulate the timescale of this dynamics. So we can describe this, capture the dynamics in terms of transition, escape from a potential well here. So we can think about one state, the up state, as one minimum of an energy potential, and the other state, the down state, as another minimum. And so there's a potential barrier separating the two. And so the transition times depend on this height of the barrier. And I can now perform some perturbation of the circuit such that I can change the height of this barrier. And so I can control the switching times between these attractors. And so I can do this perturbation in many different ways. And the way I'm going to do it here is by considering quenched disorder as an input to the network. So every neuron in this network has some baseline input from some other brain area. And now in the control condition here, all neurons have the same baseline. But now I consider a perturbed condition where I change the mean, I change this baseline value for each neuron. So introduce some quenched disorder in the network. And what that does is that destabilizes the attractor. And so it shrinks the barrier between them. And so it accelerates the network dynamics. And so now we have lower barriers and faster transition times. And if I stimulate the network now, so now I want to look at the network response to stimuli. In the control condition, if I present a stimulus to one cluster, it's gonna the cluster is gonna take some latency before activating. But what happens is that if in the perturbed condition, the barrier is lower, and so the stimulus is gonna activate, the cluster is gonna activate faster in response to a stimulus. So by modulating these barrier heights between the potential wells, the network can adjust its reaction times to different conditions. And so using this kind of idea, this is just a cartoon, but one can make it very precise in this network. Using these kind of ideas of modulation of metastable dynamics, one can explain the effects of changes of state, of internal state, on the dynamics of neural responses. And in particular, one can show that visual responses occur faster in visual cortex during running compared to resting. So when an animal is running, that accelerates the neural dynamics in visual cortex in response to stimuli. And the same happens in the gustatory cortex in response to taste stimuli. So when a taste stimulus is expected, you identify the taste faster compared to when it's unexpected. And so this is also can be described by this dynamics, this modulation of potential wells. And so this acceleration happens for small values of the perturbation, like when you are in kind of a linear regime. But what happens if the perturbations are larger? So what I want to show you in the next 5 minutes is how to use this theory of modulation of potential barrier to explain like a very interesting psychophysics law that was found 100 years ago, but it's not been explained so far. And so this is called the inverted U Yerkes-Dodson law. And so it's this very intuitive phenomenon that occurs, in everyday's life. Everybody has gone through this. So, and it's the relationship between performance on a task and the level of arousal, the level of arousal that you're in. So if you're kind of very drowsy, so we call this a low arousal state, kind of sleepy, drowsy, not very attentive, then you don't perform very well in a task. However, if you're kind of in the zone, so with just the right amount of attention, you perform very, very well in the task. That's kind of intermediate levels of arousal where you're so-called like in the flow or in the zone. However, if you're too aroused, you're kind of like very anxious, very, very stressed, super aroused, then again, you perform really bad. So there's this kind of inverted U-shaped curve that's been reported in like a gazillion different tasks in humans, in animals. This is like ubiquitous. It's just a law of psychophysics, which relates the level of arousal to the performance. And in particular, it says that at an intermediate level of arousal, there's optimal performance. And so this is work done by Leah, a postdoc in my lab. It's not published yet. We just finished the work. And so what we did is we tested this in auditory cortex now. So far I talked about motor cortex, visual cortex, gustatory cortex. Now I'm gonna talk about auditory cortex. So we have a mouse running on a treadmill, and the mouse hears some tones. And so we're gonna monitor the behavioral state or the arousal state of the animal with video cameras that monitor the pupil and the movements. And so the people found that the pupil size is a proxy for arousal. So arousal levels are proportional to the size of the pupil. So when you're very sleepy, the pupil is very small. When you're super anxious, the pupil is huge. And so in the middle, kind of in the zone, the pupil is kind of an intermediate value. So there's a lot of work on this that connected arousal to the pupil size. So what we're going to do in this task, we have presented many, many tones to the animal of different frequencies. And we record the neural activity here. So this is activity in time, neural activity in time from 60 neurons simultaneously recorded, where we deliver some tones with different colors here at different times. And then we measure the pupil, and the pupil varies a lot in time. And what we found, yes, is there a question? No. So what we found is that what we can do is we can try to decode the information about the tone that was presented to the animal from the neural activity using linear classifiers. So this is a cartoon on what this is. So we have 3 cells. So this is a 3-dimensional space of firing rates. Every time we present one tone in yellow or a different tone in blue. This is 1 point in this 3-dimensional space. And we can train a classifier to predict which tone was presented from the response, from the neural response. A linear classifier is a hyperplane in this firing rate space that separates the tones. And we can do this as a function of the pupil size. What's the performance of this classifier that's predicting which tone was presented from neural activity at low pupil, intermediate pupil, or high pupil? And what we found was that the neural activity encodes information about the tones maximally, optimally at intermediate level of the pupils, where the pupil is like in the 50th percentile here. And the encoding of information about the tones is kind of not as good, is worse when the pupil is small or when the pupil is very high and the animal was running. So there's kind of an optimal state of the network, which is this kind of intermediate pupil value. And so what we do now is we want to explain this, why this happens, using a recurrent neural network that has this attractor dynamics. So we're going to use our biologically plausible network of excitatory inhibitory spiky neurons, where we model the pupil size or the effect of arousal as this kind of quenched disorder external input to the auditory cortex. And, you know, long story short, what we found is that, in fact, this works. So what I'm showing you here on the left is the performance of the network in classifying the tones that were presented to the network as a function of the strength of the quenched disorder on the horizontal axis. And what we find here is that for small values of the quenched disorder, there's an improvement in performance. And this is what I mentioned before, the same thing that happens in visual cortex and in gustatory cortex. But for very high level of disorder, then there's a degradation in performance. So although in the linear approximation for small quenched disorder, this improves performance, for very, very large value, there's this kind of strong nonlinear relations between quenched disorder input to the auditory cortical networks and the task performance, such that there's an optimal level of disorder that's modeling the pupil size. And the explanation for this, one can explain this in the following way. So with low quenched disorder, the attractors are very stable. These are the potential. Well, there's a big barrier height separating the attractors. So the switching dynamics is very slow. And the clusters are very stiff. So they're very hard to switch. And so when a stimulus comes in, it's kind of harder to get the network into the coding state because the clusters are stiff. If we introduce some quenched disorder here in the optimal state, then the quenched disorder destabilizes the attractor a little bit, so that it makes the network much more flexible to encode incoming stimuli. However, if we introduce too much quenched disorder, then we destroy the attractor structure. And so we go into a situation where the different cluster structure is lost. And so the network degrades performance as well. So there's like an optimal state for stimulus processing, if you want, which is when these barriers are kind of still there, but small. And the attractor is in a very, very fast switching state, which is the optimal coding state. And so basically what I showed you is that we can modulate these attractor dynamics, the timing of these attractor dynamics, to explain this inverted U-shaped curve, this universal law of psychophysics, if you want, that relates task performance to the temporal dynamics of these attractors in auditory cortex. And so this explains the dynamics in auditory cortex, and in previous work, we showed that it also explains dynamics in visual cortex, in gustatory cortex. And at the first part of the talk, I showed you that it explains dynamics in motor cortex. So it seems like these attractor dynamics could be, well, and work by Remy, of course, showed that this also works in hippocampus, I guess. So these attractor dynamics might be like a universal kind of dynamical property of cortical networks, if you want. And if you're interested and want to hear more of this, I'm writing a graphic novel with the student in my lab. So the first 10 pages are on my website. We're using these ideas to model the effects of serotonin or psychedelic drugs in cortical circuits. And so we're writing a graphic novel on it. It's pretty awesome. So Audra is like an amazing artist. So how much time do I have left?
We probably started about 5 minutes late, so probably around 7 minutes.
Okay. So I'll mention the last 3-4 minutes. I'll mention this last piece of the puzzle, which is, okay, I showed you now how you generate the stochasticity in action timing using metastable attractors that explain these other contextual modulation, if you want, of behavior and performance. What about this hierarchy of multiple timescales? So kind of this is the third axis of variability in behavior. And I'm going to show you that within the same framework, one can actually explain this. So what is the experimental evidence for this multiple timescales? So there's multiple timescales in behavior, this hierarchy. Is it the same? So are there multiple timescales of neural activity? So there's been some experimental evidence suggesting that this is the case. So we can measure the typical timescale of neural dynamics using autocorrelation functions, which represents the correlation of activity at time t with activity time t plus tau, if you want. So this is an autocorrelation function. The width of this function is the typical timescale of neural activity. And people found that in different cortical areas, from the posterior to the anterior part of the brain, there's a hierarchy of timescales such that posterior parts of the brain are faster and anterior part of the brain's frontal cortex is much slower. So it's kind of a hierarchy across areas. But crucially, within a single cortical areas, across neurons, one finds a very, very a large hierarchy of time constants of this kind of autocorrelation times, varying from 10 milliseconds to 1 second or even 10 seconds of different neurons within the same circuit. So the question that we asked is like, can we generate this hierarchy of timescales in a recurrent neural network? What's the simplest neural circuits that can generate this in a robust way? And so we go back to the recurrent neural network that I showed you at the beginning. It's a recurrent network of rate units. Every unit here represents a population of neurons, like a cluster of neurons. And so we have the recurrent couplings as before. And now we introduce a new quantity here, S, which is the self-coupling of a cluster to itself that represents how big the cluster is. So this quantity S. So in a network where all clusters have the same size, so this network generates chaotic dynamics. This is it's kind of the standard model of recurrent neural networks, if you want. And so when the couplings are random and the dynamics is chaotic, if we have, this is the connectivity matrix, it's random in the off diagonal, and the diagonal part is this S value here. When all clusters are the same, then all neurons have the same autocorrelation function. So a network like this doesn't generate any heterogeneity in timescale. All neurons have the same timescale. So what we did was like, well, this doesn't work. What we did was like, well, let's take clusters of different size now. So let's allow these coefficients here, the self-couplings of each cluster, to vary. For example, if we have two sets of clusters, large clusters and small clusters here. And what we found is indeed this actually does the trick. So having clusters of different sizes generate autocorrelation functions of different width, so different temporal dynamics. And if we allow for a distribution of cluster sizes now here in the network, so many neural assemblies of different sizes, we find an exponential distribution. So this is of timescales. This is the log scale on the y-axis. So we find an exponential distribution of, or log normal distribution, sorry, not exponential, of timescales. And so we find timescales that vary over many orders of magnitude, which is kind of like what was seen in the brain. And so in particular, okay, so last comment is that one can develop a full analytical treatment of this network, recurrent neural network, using mean field theory. It's called dynamic mean field theory. This is kind of a super cool method from physics that one can adapt to. It's been developed for studying spin glasses in condensed matter theory, and one can apply it directly to these recurrent neural networks to calculate analytically these transition times. And what happens is that in the limit of large clusters, the network becomes with on and off states, just like I showed you before. This is one particular unit with metastable dynamics. And one can calculate analytically the transition times and find that the log of the mean first passage time is goes with the square of the self-coupling. So there's an exponential relations between transition times and self-couplings, such that you can generate the full hierarchy of timescales from milliseconds to minutes or hours within the same cortical circuit. So this is called the universal color noise approximation to the Fokker-Planck equation. So which we we found it's from the 80s, but now the 80s are hip again, I guess. So we used it to solve these recurrent neural networks. Okay, so then what did I show you today? So I showed you that there's this huge, three-pronged structure in temporal variability, if you want, in behavior and brain activity. At the lowest level, we encode actions as metastable attractors in secondary motor cortex. We concatenate actions into sequences using low-dimensional noise. And then we can modulate these transition times, these potential wells between attractors to achieve optimal performance in a task with this psychophysics law. And then we can generate hierarchy of timescales by changing the size of these assemblies that drive the attractors. And so this is the people who did the work. For the first part, Stefan and Ulysses, collaboration with Champalimaud. For the second part, Leah and David Warwick in my lab, and collaboration with David McCormick and Mike Weir and UofO. And for the third part, Merav Stern and Niku Istrate. This is the collaboration. And so happy birthday to Mike. And yeah, thank you for inviting me again. Yeah.
Questions for Luca?
Yeah, Luca, I have a question about the sequences. So when you have a term which is asymmetric in the GIJ, which goes from pattern mu to mu+1, and you introduce a noise zeta of t, if I remember correctly the notation.
Yes.
So I think I didn't get the distribution of zeta of t, and how, yes, exactly, and how you would think this is implemented from a biological point of view. I mean, this kind of fluctuation in the synaptic inputs.
Yeah. So thank you for the question. So, okay, so the question is in this model, okay, sorry, I'm making a mess with it. In this model of sequences, so we have the asymmetric term that concatenates attractor mu with attractor mu+1. What we did is we multiplied this with the scalar noise term, the zeta of t. And so the scalar noise term that we model here is an Ornstein-Uhlenbeck process. It works with white noise as well. And so this model, just as it looks like this, is not biologically plausible because we have multiplicative noise in front of a synaptic coupling, if you want. So what we showed in the paper is that this model is equivalent to a model where you have two areas. If you look here to the left, so one area is a recurrently connected area that's representing motor cortex, which has this symmetric part of the couplings. And there's a subcortical area, this small area here, which is not recurrently connected. It's just a area without recurrent connection that has feedback, feed4 and feedback connections with secondary motor cortex. What happens is that if you allow for noise in the connections from this subcortical area and motor cortex to be present in the discharge, if you think about this subcortical area as the thalamus, then these are thalamocortical connections, and these are noisy. If you allow for noise in those connections, then you can integrate out this subcortical area and find the effective theory for the system, which is described by this equation where the noise in these thalamocortical projections shows up as a multiplicative scalar noise in front of this asymmetric couplings. So these asymmetric couplings are integrating out this loop between cortex and a subcortical area. So these two models are mathematically equivalent. And yeah, so if you want to see the details, the more details are in the paper. Does that make sense to you?
Yes, thank you. Yeah.
Yeah, great talk, Luca. Very clear. Kind of a general question. I mean, it seems like a very natural model to a physicist. I can imagine working on it. Is there any competing class of model? Is there anybody out there that would say something different than this that you could?
Yeah. So, well, that's a great question. So there's many alternative models that explain many of the phenomena of this kind of different systems that I showed you. For example, for visual cortex or auditory cortex, there's alternative models that are not based on attractors, or they're not based on this multi-attractor dynamics. They're based on single attractors that one can modulate. And so they don't account for this variability. And so there's definitely alternative models that explain this. And so one thing that we can do now that we're thinking about is to actually find ways. So let's say one phenomenon can be explained by two models. One, which is this multiple attractor model that I show you, and an alternative model. There's a single attractor with some different ingredients, if you want. Is there a way to design an experiment that can falsify one of the two models? How can you design that? Well, we have access to a lot of these cell type specific manipulations using experimental technology. So we can control the activity of single cell types in these circuits. And so what we think is that using this is called optogenetic, for example. So we can control the activity of different classes of neurons in these models using lasers. We can turn them on or turn them off. And so we think this is a great way to actually now test this model against alternative models and see, there's going to be very specific predictions that these models do if we start to turn on and off populations at will. And so this would be a way to test alternative models. So up to it, yeah. So this is kind of something that we're thinking about. And so people have used optogenetics to test whether there are attractors in secondary motor cortex, and they confirmed the existence of attractors in secondary motor cortex by perturbing the system. This is kind of a cool experiment they did. So the basic idea behind an attractor is that if you perturb it a little bit, you go back to the attractor very fast because it's an attractor dynamic. So for small perturbation, you put it away of its potential and then it goes back. And people performed this experiment in secondary motor cortex, Karel Svoboda's group, and they actually showed that this is it behaves exactly as an attractor was supposed to do. So that was kind of super cool. So using these kind of ideas, one can now test whether there's this kind of attractor dynamics in other areas. And so, yeah, so we plan to do that with our collaborators at U of O.
Right. Okay. Thanks.
So I have a quick question.
Yeah.
So you described how these attractors chain together action sequences of different kinds. But to execute a complete motor plan, you need to be able to sort of flexibly chain together these action sequences, of course. And that chaining has to be sort of goal or task dependent. So you need to be able to kick yourself from specific attractor to specific attractor in a goal dependent way. And what's more, as we saw with the squirrels, often you'll miscue and the action sequence won't achieve what it needed to do to get to the next step. So you need to be able to flexibly say, well, stay longer. So do you have any thoughts on how that kind of thing is done?
Yeah. Well, that's a very good question. So the model that I showed you does not do that. So cannot achieve that. So the model that I show you with this so the model encodes for a single action sequence, if you want, that's represented by these asymmetric couplings. And so you can drive that action sequence or a different action sequence, but this model does not allow to flexibly, kind of decompose and recompose actions with it to achieve slightly different sequences or adapt to kind of adapt one sequence to changes. So that's not possible to do in the particular model that I showed you. But one can make a more complicated model, adding the effect of adding. So this is like, let's say, thalamocortical model for secondary motor cortex and thalamus. We need to add the basal ganglia and a controller, which would be prefrontal cortex, if you want to this model. And at that point, yes, so you are able with extra ingredients to introduce flexibility, although it's not possible to do just with the model that I showed you. Yes. So in the review that I wrote, so I showed how to do that with a, it's a more complicated cortical interaction model. Yeah.
Wonderful. Any more questions? Well, let's thank Luca. Thank you very much, Luca.